Hello, everybody, and welcome to our Precisionary Webinar scientific series. Today, we are really excited to have Professor Christopher Vaaga, who is an assistant professor from the Department of Biomedical Sciences at Colorado State University. So, Dr. Vaaga launched his research lab in 2022, following his graduate training at the Vollum Institute, Oregon Health and Science University, and also his postdoctoral work at Northwestern University. His research explores the neural circuits behind behaviors crucial to survival, with a focus on disruptions in these circuits across various disorders. And in today's talk, Dr. Vaaga is going to walk us through his lab's recent investigation into how the cerebellum interacts with the periaqueductal gray, a brain region responsible for innate fear responses. So with that, Dr. Vaaga, I want to hand it over to you, and you'll have to explain to us this wonderful image that you have on the front cover of your presentation.
Sounds good. Thank you so much. Yeah, I want to start by just thanking Precisionary Instruments for the opportunity to give a talk. I'm really excited to give a talk today. This is the first time that I'm presenting work solely collected by the trainees in my lab, so I'm really excited to share what we've been up to in the last few years. So yeah, this image was a ChatGPT or DALL-E generative AI image of sort of the title of the talk of trying to understand the neural circuits of innate fear behaviors. And so according to AI, this is what we do in the lab all day. So to get started, I want to just sort of orient you to sort of the overarching goal of my lab, which is to understand the neurobiology underlying ethologically relevant behaviors and elucidate the neural circuit mechanisms that contribute both to generating those behaviors, but also importantly, how the neural circuits generate behavioral flexibility. And so to do this, we use a multidisciplinary approach that spans different levels of analysis, including behavioral experiments, in vivo recordings of neural circuit dynamics, as well as a heavy emphasis on in vitro neural circuit function using slice electrophysiology to really try to identify the mechanisms underlying the generation of flexible behaviors. And I like this integrative approach because we can generate mutually informative hypotheses and test those hypotheses at all three levels of analysis. So as Abby mentioned, the primary behavioral paradigm that we use to study behavior, ethologically relevant behavior, as well as flexibility, is fear behaviors in mice. And so I wanted to start by just sort of defining what fear is. So the operational definition of fear that I really like was put forward by Ralph Adolphs, who said that fear is an acute intervening variable between sets of context-dependent threatening stimuli and different suites of defensive behavioral responses that are ultimately engaged in order to promote survival. And so with this definition, we can sort of place fear as this internal variable that links a variety of sensory stimuli that may indicate or signal a threat within the environment to specific behavioral reactions that are going to promote survival, such as fleeing to the safety of a nest or freezing in order to evade detection. And I really like this sort of idea that fear sits as this internal variable because it sort of intuitively makes sense, but it also provides a really powerful framework to understand how various neural disorders as well as neural circuits can change how sensory stimuli and behavioral reactions are fixed. And the example that I like to use is one that may be familiar to many of us, which is watching a scary movie in the middle of the day with a bunch of friends eating popcorn. That scary movie may not seem so scary. In fact, it may seem kind of silly. However, if you were to watch the exact same movie, so the same sensory stimulus, in the middle of the night during a storm by yourself, your behavioral reaction to that sensory stimulus might be much more fearful. And this really highlights the fact that this internal state variable of fear can be modulated by things like the context, the environment, internal state, hormonal control, as well as hysteresis or threat history. And I also like this sort of definition by Ralph Adolphs because in this context, fear becomes an evolutionarily conserved behavior that allows animals to respond to diverse threats within their environment. Now, when I talk about fear, many of you are likely thinking about Pavlovian fear or associative fear memories, in which an initially neutral stimulus, such as a tone, is paired with an aversive stimulus like a foot shock. And each of these two sensory signals are then encoded in different regions of thalamus and cortex that ultimately impinge on circuits in the amygdala in order to drive long-term synaptic plasticity within the amygdala that ultimately drives activation through the central amygdala of various effector pathways, such as the central periaqueductal gray, the lateral hypothalamus, and the paraventricular nucleus of the thalamus to drive various fear-related behaviors. However, the type of fear that my lab is particularly interested in studying are innate fears that are elicited in response to predator threats in the environment. So innate defensive behaviors are distinct from conditioned or associative fear in that they're an evolutionarily conserved set of defensive behaviors that are engaged by sensory stimuli that mimic predators in the environment. And importantly, these fear behaviors do not require any previous associative learning. And as one of the primary examples of this that we use in the laboratory is the presentation of threatening visual stimuli presented in the upper visual field to mimic either a predator flying overhead or a predator flying down towards the mouse. And as you can see in this schematic, these different visual stimuli can elicit different behavioral responses. Now, despite this this conceptual framework of an innate defensive behavior, you might be thinking that these are relatively fixed behavioral responses. But it turns out that there's an emerging set of evidence in the literature that suggests that these innate fear responses are remarkably flexible and can be modulated by different environmental conditions. And this has led to the development of what we call the threat imminence model of innate fear, in which animals will engage in different behavioral reactions depending on the proximity of the threat. So for example, using this as sort of our paradigm, a cruising predator represents a relatively distal predator that's just flying overhead, and that's going to engage a passive coping strategy of freezing in order to evade detection. However, as that threat becomes more proximal, in other words, the animal starts to, or the hawk or predator starts to fly down and try and attack the mouse, the optimal behavioral strategy is to try and run to the safety of a nest. And that represents an increase in threat imminence and a shift in the behavioral strategy employed by the prey. So because these defensive behaviors are innate, they require dedicated neural circuitry for a variety of different things that are, we think, hardwired within the brain. And so the first is the ability to detect the threat. So we think that the predominant circuit that is involved in threat detection is the superior colliculus. For example, there's a set of cells in the superior colliculus that preferentially respond to a high contrast expanding dot, basically, which would very nicely mimic an aerial predator flying down or that looming visual stimulus. Once the threat has been detected, the animal must select an appropriate behavioral response. And this is where sensory motor integration becomes really important. The animal, in order to select the best response, needs to evaluate the nature of the threat, the environmental conditions, whether there's a nest to run to. And we sort of think that these decisions are likely mediated throughout cortex. But one particular area that we're interested in is understanding the role of the midbrain periaqueductal gray in selecting these behavioral responses. And so what we know from previous work is that the periaqueductal gray is organized into these discrete rostral caudal columns, and activation of the dorsal medial PAG results in a flight behavior or a fleeing behavior, whereas activation of the ventrolateral column results in robust freezing behaviors. And so we think that maybe differential activation of these two different columns may be an important component of selecting the appropriate response. Once that response is sort of selected, the animal needs to engage in that defensive behavior in sort of a fail-proof way. When an animal decides to freeze, for example, you don't also want to have a motor program active engaging flight. And then finally, the animal needs to be able to tune or update their fear response. For example, when the sensory stimulus that engaged this defensive behavior, it actually turns out to be innocuous. And this ability to sort of update our fear responses is really important, as inflexible fear responses can result in a variety of disorders, including post-traumatic stress disorder, that I will touch on in a little bit in my talk. So our lab is particularly interested in understanding the freezing behavior of mice. And some really seminal work by Andreas Luthi's group demonstrated that bilateral optogenetic stimulation of glutamatergic neurons in the ventrolateral periaqueductal gray can drive really robust freezing in naive animals, as shown here. And also, optogenetic inhibition of the same population of neurons can reduce innate fear in an innate fear paradigm similar to what I've outlined before. And so this really suggests that the ventrolateral PAG is both necessary and sufficient to drive these freezing behaviors. However, our lab is really interested in identifying the neural circuit mechanisms that drive these behaviors within the periaqueductal gray and understanding the different factors that can modulate innate fear expression. So in order to study innate fear, we use these looming and sweeping visual stimuli. So briefly, animals are placed into an open field arena where with an overhead visual monitor that we can use to display either sweeping visual stimuli to mimic a more distal threat or a looming visual stimulus to mimic a more proximal threat. Importantly, these experiments are done in the absence of a shelter to run to, which, as you'll see, biases the animal towards a freezing response. However, when a shelter is present, the looming stimulus, being more proximal, generally drives active coping strategies such as flight to a nest, whereas the sweeping stimulus elicits more passive coping strategies such as freezing. So while the animals are exposed to these different innate fear paradigms, we video record them and then track their position in deep lab cut. And then we take that position data and convert it into a velocity trace, which here I'm showing 10 different or 20 different animals. And anytime the trace is pseudo-colored red, the animal has dropped below our freezing threshold of 2 centimeters per second. And it's important to note that as I show these traces, this gray box sort of represents our 20-second analysis window, where we'll calculate the percent time freezing. And the loom duration is significantly shorter than that period of that analysis window. However, to sort of simplify the presentation, instead of presenting the raw velocity, I'll present most of the data as a what we call a behavioral raster, in which each line here represents a different animal. And whenever the line is red, that's when the animal is engaging in freezing behavior. So I want to give you an example of sort of what this looks like. I have to switch here to out of the laser pointer view. So here's an animal in an open field arena whose position is being tracked with deep lab cut. And in the upper left-hand field here, I have an inset of the monitor in the upper visual field. And what you can see is that when we play that looming stimulus, the animal immediately engages in this really robust freezing behavior. And I promise you the video is not frozen. The animals tend to freeze, stopping all voluntary movement for about 35 to 60 seconds before they sort of break out of that and start to engage in exploration of the arena. So the first thing that I want to do is sort of outline the basic fear response to both sweeping and looming visual stimuli. So remember, sweeping visual stimuli should mimic a more distal predator threat and elicit really strong freezing responses. So when we play a sweeping stimulus, we see that animals do engage in this robust increase in freezing during this 20-second analysis window. And there's no significant difference between how males and females respond to that visual stimulus. However, when we play a looming visual stimulus, the response to the behavioral response is significantly increased, where we see this massive and prolonged freezing episodes in almost all of the mice that we record. And again, there's no observable sex differences here. And if we directly compare the responses to the proximal sweeping threat and the more distal sweeping threat and the more proximal looming threat, we see that there's a significant increase that matches the proximity of the threat. And so this data suggests that in the absence of the shelter, mice are going to engage in the best available defensive behavior, which in this case is freezing, even though in response to a looming threat, that freezing behavior is ethologically counterintuitive because it sort of sets you up as sort of a sitting duck. This data suggests that the threat imminence model that I proposed earlier isn't just encoding different behavioral strategies, but rather encoding the vigor with which the animal engages in the best defensive strategy available. So in other words, this more proximal threat is engaging a more vigorous behavioral freezing response in accordance with the increase in threat proximity. And also, we observe no sex differences in behavioral choice or response vigor, which is in contrast to some emerging evidence from Becca Shansky's lab that shows that females and males respond differently to conditioned threats. So the next question that we had is, how does threat history impact innate fear behaviors? And so to test this, we presented the same looming stimulus threat 3 times in a row during a single behavioral session, where each looming trial was separated by just 5 minutes. And what we see is that across trials, we see this gradual reduction in the mouse freezing behavior, which is suggestive of behavioral habituation over these really rapid sort of synaptic scale timelines. Interestingly, if we extend the time between stimuli from 5 minutes to 24 hours, thinking that maybe this rapid succession, the animal is rapidly learning, but in the real environment, if the animal goes to forage for food and is exposed to a threat, then the next day when they go and engage in foraging, they would likely want to maintain that fear response. So we thought that this trial design of separating the stimuli by 24 hours might prevent against this behavioral habituation that we see on short intervals. However, what we see is sort of the opposite, is that at 24 hours, we actually see what we're calling an accelerated habituation, as evidenced by this increase in the habituation between trials 1 and trials 2, and very little difference between trial 2 and trial 3. And this happens both when animals are exposed to the looming threat in the same behavioral conditions, behavioral context, or in 3 different behavioral contexts, suggesting that this isn't some form of contextual fear learning. Okay, so the next question is sort of trying to understand some of the potential neural circuits that underlie this innate fear habituation. And so the ability of an animal to dynamically regulate innate fear behaviors requires a process to integrate and update fear on a trial-by-trial basis. So, for example, that fear habituation that we saw trial by trial. It's important to note that this sort of synaptic, or this sort of ability to dynamically regulate responses must be able to occur on a relatively rapid timescale of about 5 minutes, suggesting that it might be mediated by synaptic, short-term synaptic dynamics and synaptic plasticity. So one of the main questions that we had are, what are some of the circuits that may actually contribute to this behavioral flexibility? And one of the circuits that I was interested in as a postdoc was the cerebellum. So it turns out that although the cerebellum is intimately involved in motor coordination and error correction for motor commands, the cerebellum also has an increasingly appreciated role in affective and cognitive function. And from the affective side, it turns out that the cerebellum is highly interconnected with a variety of limbic structures that support fear processing. So for example, there are direct projections from the cerebellum to the prelimbic cortex. There's also direct connections to the amygdala, the hypothalamus, and even to the periaqueductal gray itself. And cerebellar circuits are known to undergo this sensory motor adaptation during learning, which may be similarly applied to fear paradigms in which there's sort of a mismatch between the expected threat and the actual outcome of that threat. So that mismatch sort of is reminiscent of the error coding that happens in the cerebellum. And so the hypothesis then is that the cerebellum may contribute to innate fear as sort of a synaptic substrate for sensory motor learning that allows innate fear responses to be appropriately tuned and update when there is a mismatch between the sensory stimulus and the expected outcome of that stimulus. Now, there is some historical literature evidence that suggests that the cerebellum is involved in fear. One piece of evidence comes from conditioned fear paradigms, in which they demonstrated that the parallel fibers in the cerebellar vermis undergo enhanced long-term plasticity following fear conditioning that is not observed in either naive mice or mice exposed to the aversive foot shock, but in an unpaired condition. Furthermore, if we lesion the cerebellar vermis in rats, we see a reduced innate freezing response to natural predators, in this case, a cat. And that persists, so this decrease in freezing response is specific to vermal lesions and not lesions of the cerebellar hemisphere, which was an important control to make sure that the animal could still engage in freezing responses despite a lesion to a motor region. And so this data really raises the possibility that associative cerebellar plasticity might contribute to innate fear behaviors, and specifically sensory motor integration to generate that flexibility that we're observing. And so the overarching kind of guiding question that we've been pursuing in my lab is, what are the circuit mechanisms in both the cerebellum and the periaqueductal gray that generate innate freezing responses and produce this behavioral flexibility? So I'm going to start by talking a little bit about the cerebellum. Then I'm going to transition into talking about sort of how stress impacts fear responses. And then I'll wrap up by talking about some of our recent work looking at microcircuitry. So starting with the cerebellum, our question is really, what is the synaptic mechanism and functional role of the cerebellum in modulating innate fear responses? So I'm going to summarize very quickly the work that I did as a postdoc that identified a direct projection from the medial cerebellar nucleus, which importantly receives input from the cerebellar vermis, which is what has been implicated in both conditioned and innate fear in previous work. But neurons in the medial cerebellar nucleus directly project to the ventrolateral PAG, where they tend to preferentially target a population of local dopaminergic neurons. Now, the functional effect of stimulating cerebellar inputs in the PAG using optogenetics is an increase in the inhibitory postsynaptic current strength and a decrease in excitatory postsynaptic currents. And so this data, and this was mediated importantly by postsynaptically expressed D2 receptors. So this synaptic data suggested that cerebellar input to the ventrolateral PAG has the capacity to modulate the efficacy with which synaptic inputs drive activity in this VLPAG freezing microcircuit. So in my own lab, what we wanted to start to do is, if this sort of hypothesis is correct, then it could be that cerebellar activity across trials might result in this strengthening of inhibition and weakening of excitation, which would serve as a synaptic substrate underlying that gradual habituation that we observed. And so to test this, we wanted to manipulate cerebellar output in vivo and examine whether that impacts innate fear habituation. So there are a couple sort of caveats that are important to explain with this experiment. The first is that cerebellar circuits are spontaneously active, which means that manipulating circuit output is much more difficult than simply activating or inactivating, as that in itself is a signal. So ideally, we would want to sort of clamp the activity of cerebellar output so that there are no further increases or decreases. The second thing is that Purkinje cells and cerebellar nuclear cells are coupled by inhibition. So the Purkinje cells are inhibiting their targets in the cerebellar nuclei, which is just an important sort of circuit detail to keep in mind as we do these experiments. So the first thing that we did was we wanted to actually record from neurons in the medial cerebellar nucleus in vivo in order to understand how optogenetic stimulation of Purkinje cells using channelrhodopsin would impact cerebellar output. And unfortunately, what we found is that our optogenetic stimulation caused a robust decrease in cerebellar output from cerebellar nuclear cells. However, we still use this paradigm, even though it's less than ideal, as we're not able to clamp the nuclei as I proposed earlier. But our prediction, nonetheless, was that manipulating cerebellar activity will impact innate fear habituation. So to test that, we implanted a unilateral fiber optic in the vermis and optogenetically stimulated for 500 milliseconds aligned to the onset of the visual looming stimulus. And what we saw when we did this was that contrary to our prediction of an effect solely on habituation, we saw that animals with this optogenetic stimulation of Purkinje cells failed to respond to the looming threat in and of itself. And so that was a really surprising result. However, if we do look at the repetition across trials, we see that instead of showing this gradual behavioral habituation, if anything, with optogenetic stimulation of Purkinje cells, we see this sensitization. And so this data suggests that vermal Purkinje cell stimulation did in fact alter innate fear habituation, but it sort of points to this idea that the cerebellum may play a really important role in the ability of the animal to generate these freezing behaviors at all. And one of the things that we noticed when we did these experiments was that the animals sort of showed this increased anxiolytic effect. And so to test this, we did vermal stimulation in a real-time place preference assay in which whenever an animal entered the paired chamber, they received a 100 hertz stimulation for 1 second. And what we saw was that this pairing caused a rapid and precipitous drop in the percent time that the animal spent in either of these paired chambers that persisted throughout the training sessions across eight or 9 days here. And importantly, it was reversed, or when we did the reversal and made the optogenetic contingency match to the opposite chamber, what animals did is they maintained the unwillingness to enter either chamber and instead stayed in this relatively small neutral chamber, which is supposed to be anxiolytic in and of itself, given that it is made of clear acrylic. And just to sort of statistically analyze these data, I'm showing you here the response on conditioned recall day 1 and conditioned recall day 2, in which we are passively recording the animal's choice without any optogenetic stimulation contingencies. And we can see that on conditioned recall day 1, we see a significant decrease in entry to the paired chamber, and on conditioned recall 2, decreased entry into either chamber. And so this data really suggests that stimulation of the cerebellar vermis is in and of itself highly aversive. And this data suggests that the intact cerebellar circuit is necessary for normal innate fear responses and innate fear habituation. And as I just said, vermal Purkinje cell stimulation is highly aversive. So the next set of questions that we wanted to ask is to look at different physiological, environmental factors that might influence innate fear as well as behavioral flexibility. And so to do this, just as a reminder, we have this progressive behavioral habituation in control animals. And our question was, what other factors might contribute to this habituation across different trials? And one idea was that maybe a model of post-traumatic stress disorder might alter fear responsivity because PTSD is characterized as an increased fear state after either acute or chronic exposure to intensely traumatizing events. And it's just worth noting that PTSD has a highly complex etiology, but one component that we're sort of interested in is understanding whether and how corticosterone in mice or cortisol in humans might impact synaptic plasticity and freezing behaviors through activation of glucocorticoid receptors. So to assess whether acute stress or this model of PTSD alters habituation, we used an established acute stress paradigm called stress-enhanced fear learning, which is a validated experimental model to induce PTSD-like symptoms in mice and rats. And the experimental paradigm consists of 4 high-intensity, unpredictable foot shocks, and we delivered them either 1 hour or 24 hours prior to the exposure to the looming threat paradigm. And what we see is that at both time points, instead of the progressive decrease in fear habituation, we saw this, what we're calling habituation delay, in which animals maintained a high freezing response during the second stimulus and then only started to habituate on the third stimulus. And so this we took as evidence for an enhanced fear state following the cephal paradigm. So the next question is some of the synaptic mechanisms that underlie the stress-enhanced fear learning. And so to address this question, the first thing we wanted to do is to just look at whether the periaqueductal gray, and in particularly the ventrolateral PAG, expresses glucocorticoid receptors that could mediate effects from corticosterone. And what we can see in this sort of large-scale image is that the VLPAG does in fact express a high density of glucocorticoid receptors. And importantly, 1 hour after exposure to that cephal paradigm, recordings from neurons in the VLPAG show an increase in spontaneous excitatory synaptic transmission, which included both an increase in the spontaneous EPSC amplitude, as well as an increase in event frequency. When we did the same experiment and looked at inhibition, we saw that the amplitude of inhibition was unchanged. However, there was a significant increase in the frequency of spontaneous synaptic events. And together, this data sort of points to the idea that acute stress can alter innate fear habituation, indicative of an enhanced fear state. And importantly, that acute stress is altering the basics of synaptic transmission within the VLPAG, perhaps by glucocorticoid receptor activation. So this is an area of the lab that is actively expanding, and we're starting to do some experiments to test the necessity and sufficiency of GR signaling in driving these synaptic changes. The last thing that I want to talk about is the microcircuitry and synaptic dynamics in the VLPAG, and particularly whether the PAG microcolumns are interconnected, as well as examining in more detail the synaptic dynamics that occur in each column. So as I pointed out earlier, the ability to generate freezing behaviors and flight behaviors has largely been conceptualized as activation of discrete circuits in either the dorsal or the ventrolateral columns to drive these different behavioral paradigms. And so this has sort of set up this sort of paradigm in the field that these two columns sort of act independently of one another. However, a lot of emerging evidence is starting to support sort of indirectly this idea that the PAG serves as a much more important integrative sort of sensory motor integration center in the generation of fear responses. And just a couple of pieces of evidence of that is that different columns of the PAG receive widely different inputs from across different regions of cortex. And it sort of makes sense then that the PAG is sort of integrating all of those inputs and involved in decision-making and execution of the appropriate defensive behavior. However, very little work has actually looked at if the PAG is serving this sort of integrative function, whether the columns of the PAG are actually synaptically interacting with one another. And if so, what is the nature of that synaptic connectivity? And so the first thing that we did was to look at whether the PAG columns are bidirectionally connected. And to do that, we made acute coronal sections on the PAG using the compressome, and then electrically stimulated either in the dorsal medial or ventrolateral PAG while making voltage clamped recordings from the opposing PAG column. So for example, while recording from neurons in the ventrolateral PAG, we stimulated the dorsal medial PAG with a bipolar electrode. And what we saw in this experiment was that the dorsal medial PAG stimulation elicited both an inhibitory as well as an excitatory synaptic current in neurons recorded in the VLPAG. If we reversed our experiment and stimulated in the ventrolateral column while recording in the dorsal medial column, we were similarly able to elicit both inhibitory and excitatory synaptic currents. One of the things that we consistently saw across both columns was that inhibition tended to be much stronger than excitation, resulting in an EI ratio of less than 1. And statistically, there was no difference in the EI ratio between DM to VL and VL to DM. And this is just showing the difference in the stronger inhibition when we record in the VL, as well as the stronger inhibition when we record in the DM. However, what we do notice is that between columns, the strength of excitation tends to be stronger in the DM to VL direction than it is in the VL to DM, which sort of supports the idea that perhaps the DM-PAG sort of sits upstream of the VL-PAG, which actually fits with some emerging behavioral data from Tiago Branco's lab that suggests that the dorsal PAG is really involved in sort of threat processing and sort of synaptic thresholding to make the decision of when to engage, in their case, in a flight behavior. But one could argue that those same computations are being done when an animal is choosing whether or not to engage in a freezing behavior. Okay, so the next thing that we wanted to do is start to tease apart the circuitry within each column in a little bit more detail. And we started by just looking at the paired pulse ratio, while recording from a neuron in the VLPAG and stimulating in the VLPAG. So we're no longer specifically looking across columns, but rather within individual columns. And the paired pulse ratio is a commonly used metric of presynaptic release probability, and it plays really important roles in short-term synaptic plasticity. So neurons that undergo paired pulse facilitation are sort of thought to sort of act as integrators, whereas neurons that undergo paired pulse depression act in a more phasic way as sort of change detectors. And so when we recorded in the ventrolateral PAG while stimulating in the VLPAG and stimulated a pair of either iPSCs or EPSCs, what we see is that the paired pulse ratio is remarkably stable between 10 milliseconds and 500 milliseconds. So typically, if a circuit or cell is facilitating at short interstimulus intervals, you'll see a paired pulse facilitation that'll sort of taper off as the interval between stimuli increases. However, in the VLPAG, we don't see any evidence of those dynamic changes as a function of stimulus, interstimulus interval. And the same is actually true when we do the same type of recordings in the dorsal medial PAG. And so this data really suggests that the paired pulse ratio, or sort of the short-term synaptic plasticity or lability of this circuit, of these two circuits, is remarkably stable. And thinking about this data, it actually in some ways makes a lot of sense. We don't want to, the ability to generate a freezing behavior or a flight behavior needs to be sort of all or none. We don't want to have these sort of plastic changes. And so we actually think that the PAG may be very resistant to plasticity type events. In data that I'm not showing, we also found that, for example, VLPAG neurons have virtually no NMDA receptors, which obviously is then going to limit the types of long-term synaptic plasticity that they can engage in. But one of the questions that we had was, what happens if we stimulate at higher frequencies or for prolonged periods of time? Does that start to engage sort of the short-term synaptic plasticity that we're not capturing by simply stimulating twice? And so to do that, we did the same experimental setup, but now we stimulated the circuit either at 10 hertz, 50 hertz, or 100 hertz. And what we see is that in black, I'm plotting the total current. In blue is the tonic current, which is perhaps best shown here, which is sort of the slow buildup of current right before each individual stimulus. And then in red is the phasic current, which is the peak-to-peak amplitude of each individual synaptic event. So for example, from here to here is the phasic current, and this is the tonic current, and then from here to here is the total current. And what we can see is that as we increase stimulation frequency, we see this gradual increase in the tonic current that is matched by a decrease in the phasic current in such a way that the total synaptic current remains constant across the stimulation train. And this sort of matching of the tonic and phasic currents going in opposing direction results in frequency invariant synaptic transmission, which is basically the idea that we see this very stable synaptic current when we look at the total current across all of our frequencies. And that's also shown here with this slow decrease in the phasic current, especially at higher frequencies, whereas the tonic current remains consistent. And so the idea here is that the gradual depression of phasic current is matched by this increase in tonic current that results in frequency invariance. So what about the ventrolateral PAG? Do we see the same frequency invariance in the ventrolateral column? And the answer, the short answer is yes. We do see, in general, a frequency invariance of the total current. However, what I hope you'll appreciate is that the amplitude of the tonic current is significantly smaller in the ventrolateral PAG than it was in the dorsal medial PAG. So to highlight that point in a little bit more, if we actually directly compare the amplitude of the tonic current across stimuli at each frequency, we see that the tonic current in the dorsal medial circuit is significantly larger than the amplitude of the tonic current in the ventrolateral circuit. Furthermore, when we looked at these synaptic traces, what we noticed is, especially at high frequencies, the dorsal medial currents tended to have this very slow recovery back to baseline. So if you imagine that the recovery here was solely due to like a single synaptic event, you would expect the recovery would follow the recovery of the last EPSC that was elicited. However, in both the DM and VL, the recovery is a little bit slower, but that's especially pronounced in the dorsal PAG. And so to highlight that, I've peak scaled the responses to the last EPSC here in the VLPAG in black and the DMPAG in red, and you can really appreciate this increased synaptic current in the dorsal medial circuit as compared to the ventrolateral. And in fact, to quantify this, we just took the area under the curve of essentially this segment of the trace, and what we see is that the charge transfer is significantly larger in the DMPAG as compared to the VLPAG. So this data suggests that the dorsal medial PAG has this enhanced tonic current as well as this slow current as compared to its counterpart in the ventrolateral circuit. So what is the actual function of that? Does that play any important role in circuit dynamics? And so to test that, we did the exact same experiment, but now we recorded in current clamp to see whether that enhanced tonic current in the dorsal medial column contributes to a prolonged spiking phenotype. And what we saw is that just 3 stimuli at 100 hertz triggered a well-timed train of spikes, but also produced this long-lasting increase in spikes that lasted for about a second after the high-frequency stimulation. And so this is a raster plot where each tick represents an action potential as a function of time across 10 different trials from the same neuron. What you can see is that there's this massive increase in spiking in this sort of half-second to 1 second window after our high-frequency stimulation. And this data suggests that then this sort of slow current and tonic current that's produced in the dorsal medial PAG functionally contributes to a prolonged spiking phenotype in the dorsal medial PAG. And this sort of mechanistically suggests that there's some sort of a local recurrent synaptic network within the DMPAG slice that is contributing to this prolonged spiking phenotype. So the next question that we're really interested in approaching is, what is the synaptic mechanisms that underlie this enhanced tonic current as well as the frequency invariant synaptic responses? And we sort of had 2 hypotheses. We're in the middle of these experiments, but we have enough data to present one of the hypotheses, which is that it might be mediated by asynchronous release, which is sort of a feature of synapses that have expression of synaptotagmin 7. So synaptotagmin 7, or SIT7, has been identified by the Regier lab as a potential specialized synaptotagmin that is sort of built for synaptic facilitation. And the important part is that synaptotagmin 7, as compared to synaptotagmin 1, sort of remains bound to calcium for a much longer time. And that sort of ability to hold calcium means that when additional calcium comes in on the second pulse, we get more effective release. And that results in facilitation. And so in a variety of different brain circuits, Wade Regier and Skylar Jackman have demonstrated that facilitating synapses sort of lose their facilitation when synaptotagmin is knocked out, as shown here. And importantly, the ability of synaptotegmin 7 to hold on to calcium also means that synaptotegmin 7 contributes to asynchronous vesicle release, which is release of vesicles that are less well timed to the invading action potential in the presynaptic terminal. So we wanted to first test whether the stable paired pulse ratio, as well as the tonic current that we observed in the dorsal medial PAG, was mediated by synaptotegmin 7. So Skylar Jackman kindly provided the synaptotegmin 7 knockout mice. And what we see is that if we record in the dorsal medial PAG, in littermate controls or wild type animals, we see this multisynaptic peak, evident of asynchronous release. And this is not the average here, this is just a single trial highlighted. And what you can see in the synaptotegmin 7 knockout mice is that this multi-peaked phenotype is completely lost. And if we peak normalize these two currents, the average of these two sweeps, or sets of sweeps, we can see that in the synaptotegmin 7, we get this prolonged sort of smearing out of the synaptic current, likely mediated by these asynchronous release events. So this data suggests that synaptotegmin 7 is in fact in the presynaptic terminals that we're stimulating and triggering in part these asynchronous release events. And consistent with the idea that synaptotegmin 7 acts in a way to facilitate neurotransmitter release and promote paired pulse facilitation, when we knock out synaptotegmin 7 and look at the paired pulse ratio, whereas in wild type mice, we saw that very stable paired pulse ratio, in the synaptotegmin 7 mice, we see this decrease in the paired pulse ratio, especially at short intervals, suggesting that in the wild type, synaptotegmin 7 is sort of boosting transmission to sort of maintain this frequency invariant responding. And if we look at the slow current, by measuring the charge transfer after the 100 hertz stimulation, we see that there's a modest decrease in the amplitude, the charge of that slow current in the DMPAG of synaptotegmin 7 knockout mice. However, it's not significantly different from the wild type DMPAG neurons. And so this data together suggests that synaptotegmin 7 is playing a role perhaps in the frequency invariance, but is not at least capturing the whole story of what is mediating that slow current. And so the continuing work in the lab is trying to determine whether there is evidence for recurrent synaptic loops within the DMPAG that will boost synaptic transmission to mediate that slow current via circuit reverberations. And so to just conclude this component, what we've shown is that the dorsal medial PAG and VLPAG are bidirectionally connected with both inhibition and excitation. However, that connection is biased towards inhibition in both directions, but DMPAG to VLPAG has stronger excitation, suggesting that the DMPAG may sit sort of above the ventrolateral PAG. We've also demonstrated that the DMPAG has this enhanced tonic and slow current, which is likely mediated by local recurrent excitatory networks, as well as synaptotegmin 7, which contributes to frequency invariant synaptic transmission within the VLPAG to result in that sort of stable paired pulse ratio across all of the timelines or interstimulus intervals tested. So with that, I would like to thank all the members of my lab. Kate is pictured here. She was I hired her on day one. It was the best decision that I've made. She helped me set up the lab, troubleshoot how to use the compressed tome, train everyone else in the lab. She's been an absolute rock star. She participated in collecting the data and analyzing the data for the synaptic dynamics component. Jordan is a graduate student in the lab who has really spearheaded a lot of the behavioral experiments. Raven is another graduate student in the lab that's working on the cerebellum side and the RTPP experiments. And then Anna is another graduate student in the lab that's working on the acute stress as well as helping with the synaptic dynamics. And Kevin was a rotation student that also helped on the acute stress program project. And then Courtney is a postdoc in another lab that's sort of an honorary member of our lab. And I'd like to thank also our funding support and Precisionary for giving me the opportunity to present my work. And I'd be happy to take any questions.
Well, Dr. Vaaga, we really want to thank you for your talk today, taking us through a lot of your ePhys experiments. I had a few questions I'm opening up the floor as well, which is in the cerebellum, there's types of short-term plasticity. Besides the ones that you explored, how do you think that the mutation synaptic 7 would affect other aspects of short-term plasticity between different types of synapses in the cerebellar circuitry?
Yeah, I mean, so the majority of the work that we've done is looked at the
Mhm.
transmission within the PAG.
Mhm.
But there is certainly a wide variety of plasticity phenotypes in the cerebellum. Um, One of the sort of weird things is that traditional error coding in the motor program is traditionally long-term depression of parallel fiber inputs, whereas the experiments following conditioned fear, we saw paired pulse or parallel fiber potentiation. And so the mechanism behind that potentiation isn't particularly well understood, but I'd be really interested to see whether we see evidence of paired pulse, I keep saying paired pulse, short-term or long-term potentiation or depression in cerebellar
Mhm.
circuits after innate fear conditioning, or how those might change when the animal is in this background of acute stress, for example, following our PTSD paradigm. So yeah, that's definitely
Cool.
something we're interested in, but haven't had a chance to explore.
Could I ask one quick follow-up question? I sorry to
Yep.
hog you, which is related to the study of fear and startle. There's that startle response, and there's
Mhm.
synapses involved in there that goes to their brainstem. Um,
Yep.
what and I don't know the literature on this, but how based on the mutations that you're studying, how are some of those synapses and synaptic transmission affected? If I don't know if you would know.
I don't think anyone knows.
Mm,
To my knowledge, this is the first time anyone's looked at the synaptic 7 knockouts in the PAG.
perfect.
The fear potentiated startle response is a PAG mediated behavior as well. So it's very possible that the sort of effects that we have seen will impact both innate, conditioned, as well as that fear potentiated startle. But no one has looked at that question, to my knowledge.
Very exciting.
Thank you.
So I want to open up the floor, and it looks like Kaden is here, and I don't know if you wanted to ask any questions, and we have a chat question, and it is just to tell you, Dr. Vaga, that you had a great talk. So,
Awesome. Thank you.
well, thank you so much for joining us. I'm gonna reach out to you on behalf of Precisionary and our compressed tome to thank you, and so be on the lookout.
Awesome. Thank you so much.
Thank you very much. Take care.
All right. Bye.